Hi everyone, my name is Amy,
My name is Sammy.
and today we'll be going over bacterial DNA exchange, more specifically, a process called transformation. So, to start off, what is bacterial DNA exchange? Bacterial DNA exchange is the process of sharing genetic material between bacteria. In this exchange of genetic material, it allows bacterial cells to acquire new traits such as antibiotic resistance, and in some cases will allow the bacterial cells to better adapt to their environments. There are three different types of bacterial DNA exchange. The first type of bacterial DNA exchange is a process called conjugation. During this process, bacteria's pilus, or also known as the sex pistil, is utilized to transfer plasmid DNA to another bacterium. In another process known as transduction, a virus that infects the bacteria, or also known as a bacteriophage, will inject the DNA into a host cell, which is used to reproduce and incorporate the bacterial DNA into their own DNA. The bacterium's genes can then be transferred into a new cell to show off these genes.
The last exchange is transformation. This is when bacteria picks up naked DNA that is floating in the outside environment and incorporates it into its own gene. In this presentation, we will be focusing on transformation. Why is transformation important? Transformation is important because it enables bacteria to acquire new genetic traits that enhance their survival and adaptation to their environment. Scientists use transformation instead of conjugation or transduction because it's simpler, faster, and easier to control in a lab setting.
Now we'll be going over the different steps of transformation. We'll first start off with competence.
In the competence stage, the bacterial cell becomes able to take in foreign DNA through methods like heat shock or electroporation. These methods temporarily make the cell membrane more permeable so the DNA can enter.
In the next step, it is known as DNA uptake. Once a cell dies, they break apart and release their DNA into the environment. This allows bacterial cells to scavenge through their surrounding environment and to pick up this now called free-floating DNA. Free-floating DNA or naked DNA binds to the receptor proteins of the bacterial cell and is brought in through a channel.
Next, we have integration. Once the DNA is inside, the integration stage begins. The foreign DNA can either combine with the bacterial chromosome or remain as a plasmid inside the cell. This allows the new genetic material to be maintained and passed on when the cell divides, giving the bacteria new traits or abilities.
In the last step, it is known as replication and inheritance. This is when the new genes are activated and give the bacteria the new traits such as antibiotic resistance. The inherited traits will spread within the bacterial population. This is a visual representation of transformation with DNA fragments. We can see that the DNA fragments are represented by the purple strands, meanwhile the bacterial chromosome is represented by the blue ring. Only some bacteria are naturally competent, which means the DNA fragments can attach to the cell membrane. Once this occurs, the bacterial cell has uptaken the DNA.
When foreign DNA enters a bacterial cell, a few things can happen. On the left side, labeled integration by non-reciprocal recombination, the foreign DNA is inserted into the bacterial chromosome. This DNA becomes part of the cell's genetic material, so the transformation is successful. On the right side, labeled degradation, the foreign DNA cannot integrate and is broken down by the cell. In this case, the transformation fails.
Now we'll be going over limitations of transformation.
One major limitation of transformation is that bacteria can accidentally take in harmful or unwanted genes, such as antibiotic-resistant genes, found in the environment. When this happens, the transformed bacteria can spread these resistant genes to other bacteria through gene transfer, which makes infections harder to treat and poses a serious public health risk. Another limitation is that not all bacteria cells are capable of naturally picking up free-floating DNA. Only certain bacterial cells are naturally competent, causing scientists to use artificial methods to make the bacteria cells temporarily permeable. This allows non-competent bacteria to successfully take in foreign DNA.
The last limitation of transformation is the intake of disrupted genes. This can cause mutation, which will cause the bacteria to multiply. Thank you.
Thank you.